All right, with that, I'm going to get started. Welcome to the Immunology Breakout Session. My name is Stephanie Sprenger, and it's my pleasure to chair this session here on immunology at MIT. What is immunology, and why do we care about it in health and disease? So with that, I would like to give a very brief introduction on what the immune system is and why we care about it when we think about physiology as well as disease states. The immune system, and this is a slide from class, can be very simply depicted as a seesaw. And this is a very simple oversimplification, where the immune system is trying to balance fighting pathogens, but also avoiding to attack the body itself. However, as I just said, this is a very, very drastic oversimplification of what our immune system is actually doing. So this seesaw is probably more accurate, because we are not living in isolation, and our immune system is not acting in isolation. Lifestyle, diet, stress, all of these factors affect how our immune system is working, and diseases push our immune system every day to react, and our immune system still has to keep at bay, not attacking ourselves. And an imbalance within the immune system can cause diseases, but likewise allow the resolution of diseases like pathogenic infections. And it can also affect our health and our well-being, and likewise, our well-being, being happy, will affect our immune system. Now, this is a human body depicted as dots. One dot is 10 to the power of 9 cells, and one large square is 1,000 dots. Now, how big is our immune system if this is our entire body? You see here in red and in dark red the two major subtypes of cells that make up our body. The light red cells are erythrocytes. These are bone marrow-derived cells that carry around our oxygen. They also have some level of immune function. The dark colored cells are all bone marrow-derived immune cells. So these are the cells that fight pathogens, fight cancer, but also are responsible for inducing autoimmune diseases. And as you can see here, in contrast to the other organs, our immune system is, by and large, the largest organ in our body. It is likewise not a single solid organ. It is disseminated throughout our body. It has a network of nodes, hubs, meeting points called lymph nodes that are spread throughout, and this is where our immune cells actually communicate with one another. And our immune system can be very broadly subdivided into different elements. We have the innate immune system, and this is the part of the immune system that senses if there's danger somewhere. And then we have the adaptive immune system, and specifically two arms, the cellular arm of the immune system and the humoral arm of the immune system, mediated by T cells and B cells respectively. And these are the cells that instill memory in our immune system. Meaning if you get a prophylactic vaccine against flu, these are the cells that memorize that they've seen flu before and are better at eliminating the virus once you see it. These two arms of the adaptive immune system act slightly differently. The T cells recognize specific patterns on cells and eliminate those cells, versus the humoral arm is mediated mostly by antibodies. And throughout this session, we will hear from experts across all of these areas of our immune system. Specifically, Facundo will talk to us about the humoral immune system. Arup, later on, will talk about B-cell responses. Jessica will talk about the innate immune system and how patterns are hardwired. Hari will put many of these contexts into how cells interact with one another. And Michael, at the very end of the session, will talk to us how we can use those basic principles and use it to engineer our immune system. This is the summary of these 5 professors' titles, which I won't repeat for time constraints. And I'm really excited about this session. And with that, I would like to welcome Facundo Batista as our first speaker to the stage.
Yes, good morning, everyone. As Stephanie said, my name is Facundo, and yep, there we go. And that is a typical Argentinian name, and that is why I'm going to be speaking with this strange accent all the way. And so When people is asked about what has been the best biotechnology discovery in history, many will argue that it will be wine or beer. I and many others will argue that it's going to be vaccines. And essentially, vaccines has been the source of protection over the last just 5 decades and saving life for about 150 million human beings. And although we know that vaccines work, we don't know how they work, but we know that these molecules, the antibodies that are able to bind pathogens and trigger function, they are the main responsibles of vaccine protection or a correlation with them. And antibodies are not responsible of vaccine protection, but they are also critical in terms of treating disease. And I am using this example just to point out that in giving an antibody to a person, an antibody that recognizes the malaria parasite, it will protect that person for 1 year just by injecting the antibody into them. And in these antibodies has been the base of many therapeutics cures to cancer and infectious disease. And as such, it makes them a key target for bioindustry. And the industry of antibodies is at the moment in the order of 300 million a year, and is estimated to be in $1 billion in about 10 years. That is why we are particularly interested in trying to understand how these antibodies are generated. And in this regard, antibodies are produced by a particular type of cells that are plasma cells. And these cells are really antibody factories that they produce millions of molecules per hour every single day. But plasma cells are the result of a particular pattern of differentiation of B cells, and antibodies initially are produced on the surface of naive B cells, and those cells are able to recognize either viruses or either antigens that they are given through vaccination. And once they do that, they will do something rather unique. They will get together and start to divide, and they will form these that are in structures that are known as germinal centers. And here they will do something magic. What they are going to be doing is they are going to mutate the original gene that carry that protein in order to make that antibody much more potent over time, and this antibody will become much more specific for a particular antigen or a tumor. And the way that this happens is that initially, when we get vaccinated or infection, we produce very low affinity or not high quality antibodies. But then because of the germinal center formation and this specific maturation that I mentioned, the antibodies gain high affinity and specificity over the differentiation of the cells that then relies into kind of the long-term survival of this antibody production. And just to give you an idea, we all being vaccinated to measles here, and those antibodies those plasma cells that are generated in the very early stage of our life, they will live with us for about 8 years until we pass away. So my lab, what we do is we ask fundamental questions about how B cells, biology, and antibody responses happen. And indeed, I will show some examples and one particular discovery that we did that really kind of enabled us to do really absolutely cutting edge. And this is, what we have been doing over the years is to manipulate or try to create humanized mouse. In this mouse, what we do is we change the genetic code in the different chromosomes to now express B cell receptors or antibody on the surface of naive cells that are not any longer mouse, that they are humanized. And as such, we can study the immunoresponse of these mice as these B cells will be practically human. So we use this technology to develop and unravel several key questions in terms of B cell maturation and biology. And one of them was, for example, this is how a plasma cell looks. And as I told you, these plasma cells live with a very long time in our bone marrows. And these plasma cells are able to produce about 100 million molecules of antibody per cell per hour. And they need to produce that over the course of many, many years. And in order to do this, what we show, in particular a PhD student in the lab, Stephanie, Sophie, what she has done is she has particularly looked into the code of translation of that protein. For those of you who are not familiar, proteins are expressed as an RNA. They are translated by this adapter that constitute the amino acid or bring the amino acids to them, that they are the shape of the protein. And what Sophie found was that these tRNAs are critical in programming the plasma cell to produce this large level of antibodies there. We don't only look into plasma cells, we also look very much into what antibodies do into our response. And I know Arup is going to talk a little more about this, but essentially what we show is that we try to represent this as a castle. Germinal centers that I mentioned to you before, they exist in the body, and antibodies that we produce, they are able to block the entrance of particular cells or to facilitate the entrance of others. But we don't do only B-cell biology, but we try to understand how we can translate this B-cell biology and knowing in antibody responses to try to inform vaccine design and offer novel therapeutic development. And in this, I will give two examples of those. We have used this, what we call this mouse machine that I mentioned to you, mouse that produce antibodies that look like humans. And what we have shown through immunizing or vaccinating those mice, that we can produce much more potent antibodies that they can now protect us to malaria. And this is of particular function because, or particularly important, because if we have an antibody that works much better than another, that decreases the cost of the drug by several orders of magnitude. Similarly, we have been involved in a large consortia to try to use this humanized mouse model to characterize different antigens, and this is the surface of HIV, and these are different antibodies binding to different portions of HIV. And in large collaboration, what we have been using is our humanized mouse models to enable clinical trials for the production of HIV vaccine. And in that regards, in particular, we have taken these immunogens to the clinic, and one clinical trial is going to start in next January, and this relates in particular to this portion of the protein and the production of antibodies against that protein, or another ongoing clinical trial together in collaboration with Moderna that is particularly focusing in how we can develop antibodies that recognize this part of the protein of the virus. So with that in mind, actually, I would like to introduce the next speaker, that is Arup Chakrabarty, and he will be digging more into the B-cell response and the biology of it.
Thank you, Facundo. So as Facundo mentioned, it is estimated that vaccination has saved about 150 million lives in the past 50 years. But we don't have effective vaccines against highly mutable viruses. For example, we don't have a universal vaccine for the flu. So every year we guess which strains may evolve and design a new vaccine. And the situation is similar now for SARS-CoV-2, which causes the COVID-19 disease. The way to better confront these scourges is to design universal or variant-proof vaccines. And a part of our work is focused on trying to address fundamental scientific questions which, if answered, would aid the pursuit of this quest. And toward this end, we bring together physics-based computations and machine learning, along with tests of predictions in animal models and clinical data. These latter efforts are carried out in collaboration with basic and clinical immunologists. And for the vignettes that I shall highlight today, the collaborating labs were led by these individuals. Now, as Facundo mentioned, upon infection or vaccination, antibodies are generated by a Darwinian evolutionary process that occurs in germinal centers or GCs. Herein, B cells evolve by mutation and selection to bind more strongly to the antigen which is displayed in these GCs. And that is why, as time ensues, the memory cells and plasma cells and antibodies that you generate become more potent. Now, upon re-exposure to the antigen, the existing memory cells are first rapidly expanded to create a wave of antibodies that is meant to confer protection. But new GCs also form, but over much longer timescales, to generate more memory B cells and antibodies. Now, the first vignette that I will describe stemmed from our asking why 3 shots of the COVID vaccines protect better against a highly mutated variant like Omicron, while 2 shots do not, even though all 3 shots encode for the Wuhan original strain of the virus. So this Darwinian process is driven by antigen displayed in GCs. How does it get there? circulating antibodies bind to the antigen and deposit it there. Now, when you get your first shot, all you have in you circulating are generic, weakly binding small numbers of antibodies that bind to very little antigen and deposit them there before the antigen degrades. And you can see this by imaging lymph nodes of monkeys after the first shot. And here you see there's hardly any antigen there. So if you think of the antigen as the prey and the B cells as the predators in this Darwinian process, when there's very little prey, only the best predators are going to survive. And that is why, after the first shot, the antibodies and memory cells that you generate are those that initially bind strongly to the spike of the virus and bind to regions called immunodominant. And here I have depicted these antibodies binding to the spike of SARS-CoV-2. Now, after the second shot, the corresponding memory cells are rapidly expanded to generate antibodies. Now, the regions that are in red, they are mutated in Omicron compared to the Wuhan strain. And you see they are in the binding footprints of these antibodies, and so they're not very effective against Omicron. But after the second shot, you also form new GCs. But now you've got circulating antibodies that are antigen-specific, and say they bind to and deposit lots of antigen in the GCs. And you can see this by imaging the monkey lymph nodes after the second dose. You see there are lots of antigen. Now when there's lots of prey, the not so good predators can also survive and evolve. And so antibodies that bind more weakly initially to regions called subdominant that are not mutated in Omicron can evolve. And this effect is further amplified because these antibodies that bind to these immunodominant regions can enter GCs, bind to them, thereby masking them and effectively elevating the amount of these subdominant regions that are present. So now the memory cells that bind to these regions that are not mutated in Omicron that you've created are rapidly expanded and become antibodies that are protected against SARS-CoV, against the Omicron strain. Similar mechanisms are at play for our demonstration that instead of giving the vaccine as just one shot as a bolus, as is normally done, if you distribute the same amount of antigen over 7 days in 7 doses over an escalating pattern, then you get lots more antigen than just the bolus, which leads to a more potent antibody response compared to the bolus in mice. And very recently, this has been recapitulated in a human clinical trial. So from computational biophysics to the clinic, if you like. But 7 days, 7 shots is not practical. And so by better understanding these feedback loops, we have now designed a 2-shot regime that puts a lot of antigen on the surface in the GC, and you get potent responses, just like the 7-day one. We are now trying to explore how similar mechanisms can be leveraged such that antibody therapy can potentially lead to a cure for HIV. And finally, let me note that we have computationally designed nanoparticles that display spikes of diverse sarbecoviruses, the virus family from which SARS-CoV-2 is derived. And this is the potency of the antibody response in mice to several sarbecoviruses that may spill over from animals. And you see that our computationally designed one is more potent than the state-of-the-art nanoparticle and much better than the bivalent vaccine that many of us have taken. Thank you for listening. And our next speaker will be Professor Jessica Stark.
Thanks and good morning, everyone. It's a privilege to be here and to share our group's perspective on understanding and engineering immune responses. I want to start by first affirming your choice to come to this breakout session because we really are in a very exciting time for immunology in the midst of an immunological revolution in medicine. We now understand that there are a wide variety of human diseases and conditions that can be effectively treated by harnessing the immune system. As we've heard from Arup and Facundo, we've been able to use vaccination to prevent or even eliminate infectious disease by teaching the immune system to recognize and respond to pathogens. In cancer, we've been able to use immunotherapies to elicit decades-long remission from disease, really the closest we've come to a cancer cure. And nevertheless, we remain limited in the kinds of conditions that we can treat and the number of patients that we can help with immunotherapy. And so we still need new paradigms to harness the immune system if we're going to realize the full potential of immunology for human health. We see an opportunity to target cell surface sugars called glycans as an untapped axis of regulation in the immune system. Every cell in your body, and in fact every cell on Earth, is coated with a thick layer of glycans. And these glycans exert influence in nearly every immunological process. Glycans can directly control immune cell function. They can activate or inhibit immune cells by binding to receptors on immune cell surfaces. They control how immune cells move throughout the body. They can help immune cells exit the blood and home to sites of inflammation or infection in tissues, helping your immune system locate and eradicate disease throughout the body. Glycans play important roles in innate immunity, where they help your immune system do one of its most important jobs, which is to distinguish self and non-self, telling the difference between host and pathogen. And they can further influence adaptive immune responses. For example, by helping determine whether an antibody will elicit a pro-inflammatory or an anti-inflammatory immune program. Despite all of these examples, however, of how glycans play important roles in the immune system, we have lacked the tools to comprehensively study this biology and to target glycans therapeutically. And these are the gaps that my lab, which opened here at MIT about a year ago, seek to address. We are building platform technologies to help us fully understand how glycans regulate immune responses and to translate these biological insights into the design of new kinds of immunotherapy. I want to share an example of how we've been able to target glycans for cancer immunotherapy that builds upon recent fundamental and translational advances in the field. In order to grow and spread throughout the body, cancer cells upregulate molecules on their cell surfaces that bind to inhibitory receptors on immune cells. By engaging these so-called immune checkpoints, the cancer cells can put the brakes on the immune response, allowing them to evade normal immune surveillance. However, if you block these immune checkpoints with something like a monoclonal antibody, there's the potential to reprogram and reactivate the immune system to sense and respond to cancer. These checkpoint blockade immunotherapies were tested initially in metastatic melanoma, an incredibly aggressive form of cancer. And in one of these trials, patients who received a combination of therapies targeting the PD-1 and CTLA-4 immune checkpoints, about 30% of them mounted a robust immune response to their cancer that basically stopped the disease in its tracks. These patients' diseases had not progressed in over a decade following the start of treatment. And in light of the success in melanoma, checkpoint blockade immunotherapies have now been tested in a variety of other cancers and are now approved to treat about 50% of cancer patients. However, across all cancer patients, only about 20% benefit from these therapies. And so now the question is why? How are cancer cells able to evade immune surveillance and resist immunotherapy in most cancer patients? And that's where glycans come back into the story. It turns out that cancer cells dramatically alter their cell surface glycosylation. This is a hallmark of cancer that's been observed in many different tumor types and across many different patient cohorts. And we now understand that one of the reasons that cancer cells remodel their surfaces in this way is to engage a class of inhibitory receptors on immune cells that are called lectins. So when lectins bind to these glycans on cancer cell surfaces, they trigger inhibitory signaling pathways that shut down immune responses through the exact same mechanism that occurs when the established immune checkpoint receptor PD-1 binds to its ligand PD-L1. And while we have a litany of drugs that target the PD-1, PD-L1 immune checkpoint, we have no approved therapies to target these glycoimmune checkpoints. We recently developed a new kind of molecule that makes it possible to target glycans for cancer immunotherapy. These molecules we're calling antibody-lectin chimeras, or ABLECs, and they're bispecific. One half of the molecule is an antibody domain that can target specific cells in the body, for example, cancer cells. The other half of the molecule is what we call a decoy receptor. It's the binding domain from the lectin receptor we want to target, which serves to bind and block the same kinds of glycans that would otherwise inhibit immune responses. And so our hypothesis was that with ABLECs, we could block these glycoimmune checkpoints and elicit more potent anti-cancer immune responses. We tested these therapies initially in vitro with primary human immune cells like macrophages and NK cells. These are two different kinds of innate immune cells that we could imagine activating with our ABLEC therapy. And you can see that for both macrophages and NK cells, treatment with our ABLECs elicited more potent cancer cell killing compared to a monoclonal antibody control. And in vivo, in a humanized model of metastatic cancer, we observed that animals treated with our ABLEC immunotherapy exhibited reduced tumor burden compared to animals treated with the control molecule. With this proof of concept, we launched a new biotechnology startup called Velora Therapeutics that aims to bring ABLECs to patients. The exciting thing about the ABLEC platform is that it's modular. You can imagine mixing and matching different antibody and lectin domains to enable a variety of different therapeutic mechanisms of action. And Velora is leveraging this modularity to develop therapies, ABLEC immunotherapies for indications in oncology and immunology. But this is just the beginning. Glycans play pivotal roles in the immune system and are really attractive therapeutic targets. My group is developing the platform technologies we'll need to explore this emerging biological frontier and enable both fundamental and translational advances. So with that, I'd like to thank my lab who make this work possible. I look forward to taking your questions, and I'd like to invite our next speaker, Professor Hari Wong, to the stage.
It's great to be here. I want to talk to you a little bit about some of the complexities that exist in studying the immune response, why they exist, and how our group has been developing technologies and implementing technologies to try to overcome them. We spend a lot of time thinking about how the immune system operates in its native environment. So not in a dish, not in blood, but actually where it needs to function in tissues. And the reason we do this is at least threefold. One is that the immune system is a system of cells. It's not one cell, it's multiple cells. And these cells typically form multicellular units of control. They assemble in space and in time into intricate structures that do things that no one cell type can do on its own. You get emergent dynamics, emergent functions, and it's very hard to reconstruct these units in vitro settings. The second point I want to make is that the immune cells within these units are communicating dynamically. They're engaged in physical interactions with one another. They secrete molecules over short distances and over long distances. And the communication is always evolving over time and in space. And lastly, the point I want to make is that the most interesting aspects of the immune response, at least in my mind, both in physiological and pathophysiological settings, are not mediated by the bulk of the cells. They're typically mediated by rare outlier cells on the ends of the distribution. I'm not sure how to go back. There we go. So let me walk you through technologies that my lab has been developing to try to overcome each and every one of these three challenges. We typically take an imaging-based approach to really be able to resolve and study these cells in their native environment. So what you're looking at here on the left is a lymph node that lies downstream of the kidney. I want to direct your attention to this inset over here. We developed multiplexed imaging technologies using antibodies that now allow us to visualize anywhere between 20 and 60 proteins directly in tissues where they where the immune response is occurring. What you're looking at here on the zoomed in inset is a multicellular unit that's controlling T cell responses. You don't have to worry about what all the colors are. The point I want to make is that there are innate immune cells here, like Stephanie introduced earlier, that are initializing T cell responses. There are these green cells here. These are regulatory T cells. There's not one of them, there's many of them. These are cells that are trying to dampen the immune response. And there are conventional CD4 and CD8 T cells here. These are cells that are trying to promote inflammation and promote the immune response. And if you try to study any one of these cell types alone, you can't really figure out what's going on. It is the integrated behavior of all three of these cell types in space and in time that is predictive of how the immune response will behave. So we can address this. We can identify the units of control. We can identify the relevant cellular players. What about the communication aspect? Well, we've now developed ways to preserve tissues in a manner that allows us to estimate length scales of communications between cells. Let me walk you through what I mean. Here, outlined in white, these are two endogenously activated conventional T cells. They are producing a molecule called IL-2. This is a cytokine that's shown in red. This cytokine will get secreted and diffuse in space. These surrounding cells in magenta, these are regulatory T cells. Like I said earlier, these are cells that try to dampen the immune response. And in green, we're reading out signaling downstream of the IL-2 receptor by measuring the phosphorylation of STAT5, a transcription factor. If we look to the right, what I want you to take away from this is that there is a gradient of green. And it's largely in the regulatory cells here. And by reading out this gradient, we can infer how far the cytokine must be diffusing in space. And this is important because cells that are positioned here, close to the producing cells, are going to see a very different concentration of this cytokine than cells that are positioned way back here. And therefore, cells that are positioned closest to the producing cells are going to adopt a different fate than cells that are positioned way back here. So this is a source of variation, how the immune system generates heterogeneity in its responses during a dynamic immune response. This is all well and good, but it's fixed. These are static images, and the immune system is not static. It's dynamic, as I told you earlier. Communication is dynamic. So we've also invested in technologies that allow us to peer inside of living organisms. This is a mouse, and actually watch the immune system in real time. What we've done here is a small surgery in a mouse that's under anesthetic, and we're looking behind the knee. It's the popliteal lymph node, and we're peering in and we're watching the immune response in real time. This is it doesn't really matter what's going on here, but the cells in magenta, these are the regulatory T cells that are dampening the immune response. They're not fixed. They're zipping all over the place in real time. And the cells in magenta, these are conventional cells, conventional T cells that are trying to initiate an immune response. So there's a complex choreography that's always going on in our bodies at any moment in time. Now, what about the last point I raised? How can we identify the rare cells that are doing the most interesting biology? The technologies I showed you so far are fantastic, but they're limited in the depth of tissue that we can image. We can't look at enough volume with those methods. So we have to take a different approach, and the approach we use is an optical clearing method that allows us to treat our organs of interest with a chemical concoction that homogenizes its refractive index. What that means practically is that it turns our tissues from completely opaque to clear and transparent. And from a microscopy perspective, this allows us to now image the entire organ in three-dimensional space without any light scattering. Here's an example of that. This is a mouse uterus that's been cleared, and we're going to zoom in. The cyan, these are the uterine glands, and what we can find are these rare clusters of T cells here in magenta that are wrapped around the glands doing some type of important function. That's very distinct from the rest of the cells in the entire organ. These are the rare cells in the tail of the distribution, and they're functioning as a unit. But again, it's very different behavior than the bulk of the distribution. So I've shown you the technologies that allow us to overcome the challenges, but it's not good enough for us to just take pretty pictures in movies. We need to combine these experimental methods with computational approaches that allow us to extract as much useful information from our movies and images as possible. So for that, we've invested in computational algorithms that allow us to define the boundaries of individual cells and reconstruct our organs of interest with all of their features and protein expressions we can do this in the computer. So what you're looking at here is a computational reconstruction of a lymph node where every dot represents an individual cell, and we can analyze specific regions of space, which is important because different reactions occur in different places. We can use these measurements to then conduct a variety of quantitative types of analyses, similar to what you would do in data science. We can take these measurements and parameterize mechanistic models using systems of equations to simulate how different aspects of the immune response will proceed. We can make predictions, and then we can go back in to model organisms and test those predictions using genetic or environmental manipulations. And through these types of technologies, I think this is how we'll get a better understanding of fundamental operations of the immune system and understanding how different therapeutics actually work in their sites of action, in their tissue sites of action, that is. So with that, thank you, and I'm pleased to introduce our next speaker, Professor Michael Birnbaum.
All right, thanks to Hari, and it's good to see you all today. So in my lab, we want to be able to see what T cells see. So just to give you a sort of a graphic I pulled from a recent review. When a T cell is looking for cells that are infected or otherwise transformed by cancer, the way that this actually works is that every T cell has its own protein sensor called a T cell receptor, which takes recognition of what we call an HLA or an MHC molecule, which presents a short peptide. And these MHC peptide complexes present a census in terms of what the cell is expressing at any time. So when a cell is expressing just the normal proteins that make a hepatocyte or a lung cell, then T cells leave the cell to go about its business. When a cell is otherwise altered, so whether it is has an intracellular bacteria, infected by a virus, or transformed in terms of becoming a cancer, then the peptides are different, and the T cell is able to recognize it through the T cell receptor and then kill the cell, among other immune functions. So for me, one of the fascinating things about this is that this actually serves as numerically sort of an interesting thing to think about. So in each one of us, and my math could be off by a factor of 10 in any direction, but there's roughly, say, it's sort of around 50 to 100 million unique T cell specificities floating around in each one of you at any given time, out of a possible 10 to the 15 possibilities for T cells, for just combinatorically for the receptor. And these are recognizing these peptide MHC complexes where, in terms of what you theoretically could have in you, again, math is back of the envelope, but something around 1 trillion possibilities in terms of peptide and MHC combinations in any one of us. They're not all present at any given time, but there's no way for the immune system to know which one of these you may encounter during your life and which ones are purely theoretical. This is all happening in a system that has to be incredibly sensitive, right? Because if you're infected, you have to be able to recognize it at what we would consider incredibly low affinities, so interactions that occur at body temperature that last for maybe about a second. And if you do the bookkeeping, it turns out that each one of these T cell specificities would have to be able to recognize on the order of 100,000 different antigens in order for the books to balance here. Otherwise, you could have, on one hand, for example, a pathogen which is able to completely evade detection. And somehow, on the other hand, you have to be able to do this in a way where you still have enough specificity that the recognition of a virus doesn't inadvertently lead to the recognition of any of your own healthy tissues. So your immune system is doing this every day, all day, for each of us, very nearly perfectly, all the time. And I really want to know how this is able to work, and able to work, in a way that is so specific in order to allow this to happen. Understanding this specificity is going to help us understand biology. It's going to help us create a next generation of therapeutics, and increasingly, especially with the explosion of progress in AI, the ability to make better computational tools. So for all of this, though, what we remain limited in is the number of examples which we've been able to really observe. So this is a field, understanding how T cell receptors recognize peptide MHCs, that has really been sort of in progress as long as I've been alive. But we've studied really relatively small numbers of examples. So in order to be able to make progress, we're going to need to be able to look at much more data. So in my lab, we want to develop better tools in order to be able to understand this. And so we think sort of quite broadly about how to do this. We use a variety of approaches. I want to tell you just for a couple minutes about one of them that we've done in our lab. And what we've actually done is repurposed a common laboratory tool, which itself is a repurposed virus. So we work in our lab with lentiviruses, which is a heavily engineered for laboratory use, safe version of a virus that started as HIV, of all things, where we re-engineered it to say, okay, what if we take these lentiviruses, which are commonly engineered to infect any cell that you might want them into in the lab, and have them infect only T cells by putting peptide MHC molecules on their surface. So in this way, you create a virus that is specific for a given T cell. And lentiviruses are quite unique because what they are able to do as part of their life cycle is leave a permanent record of their infection into a cell's genome by incorporating the viral genome into the host genome. So our thought was, if we put a peptide MHC on the virus, we could have a system where we have infection mediated by the molecule on the outside with a record kept by the molecule on the inside. And if we're able to build large collections or libraries of these as a tool, which is common in biotechnology, we could use this as a recorder of this peptide MHC T cell receptor interaction. So we set about doing this a few years ago, and we were able to show that this actually works. So you could create a virus with a peptide MHC. If that virus encounters any cell that doesn't have the right T cell specificity, even one that's close but not exactly right, you see little to no infection. So this is a virus encoding cells to turn green, very few green. Where if you have a virus that does have that match, you get robust infection of cells. So we created a tool, a method which we call RAPTOR, receptor antigen pairing by targeted retroviruses, where we're able to create libraries of these viruses, mix them with T cells, and then allow for the mixtures to only infect the right combination, so the mixture of viruses only infecting the correct T cell. We're able to then isolate them and use single-cell sequencing in order to get the information out of what the T cell receptor is and what the antigen is on a many versus many basis, so something that we are trying to approach the scales of how the immune system itself would work. And we're able to show that this is able to work, that you're able to identify, even in a complex mixture of viruses with different peptides, once you sequence the viruses that are able to infect, you're able to get the right answer back out. This is a tool we're very excited about, but a little belatedly, we realized that if you're able to infect a cell in order to deliver a genetic cargo for something like a fluorescent protein or a barcode, that you should also be able to do it to deliver some type of therapeutic cargo as well. And so we're in the process of exploring this too. And the way that we're thinking about this is in terms of different types of cell therapies. So it turns out that your immune system can mount a defense against tumors all on its own. We call these tumor-infiltrating lymphocytes. This is something that often is not effective on its own, but is something where just recently, in the past few months, there's been a clinically approved therapy where these cells are taken out of the tumor, grown back up, and reinfused. So this is great because it's recognizing exactly what happens to be in your tumor, but is tortuously complex to make. Correspondingly, we're able to engineer T cells through something called a CAR T therapy, where we're able to point T cells in the right direction, but this is also complex to make and only works for certain cancers. So what we've done in our lab is created the same viral system where now instead of delivering a barcode, we're able to deliver therapeutic cargoes to the T cells. So we're able to, even in vivo, infuse a virus which is able to find the right T cell, reprogram it genetically to become more immunoactivated in one way or the other, and then able to extend lifespan in mice. So we're in the process of exploring sort of both of these aspects in our lab, and this is where the sort of collaborative and big picture thinking of MIT really comes in handy. So on one hand, we are a part of, and I'm the leader of a team funded through a mechanism called a Cancer Grand Challenge in order to truly try to generalize the predictions of peptide MHC molecules and T cell receptors to design these receptors. So this is a team of immunologists, clinicians, engineers, all working together, so several of which at MIT, but also for individuals around the world, to really try to address one of these large problems in immunology. How can we make these predictions? And then we're also working to take our targeted viruses and turn them into a drug in a company called Colonia Therapeutics, where we're able to show really promising data that's been talked about at meetings, both in terms of mice and monkeys, with the hopes of starting a clinical trial in the next year or so. So thanks for your attention. I believe we're moving to a Q&A session.
Please join me in thanking the speakers again, and I would like to invite all the speakers on stage. And this will be an open Q&A panel, so if you have questions, please raise your hand and we will call on you. But I would like to start off this panel by getting each one of your takes on why immunology at MIT is different to normal immunology departments that are typically housed in medical schools and are very embedded into clinical practice. Facundo.
Well, MIT is an unique ecological environment where you get direct access to engineer and computation that doesn't occur generally in a normal immunology department. So I always train in institutions that they were kind of having a large breadth of collaboration, and particular for me, I can talk, is rather a unique opportunity to be close people to do things that I'm not an expert on.
Well, as Harry mentioned, the immune system is a stochastic, dynamical system with many participating components in a fluctuating environment that work together to ultimately give you a coherent response, either to pathogens or to cancer, or sometimes when it's aberrantly regulated, to yourself. And if we have to understand that, we have to bring together tools from basic biology, from engineering, computation, and systems design in order to really make that happen, and then translate that into therapies and vaccines, et cetera. And MIT has all these components, from basic biology to systems engineering, and it's an inherently collaborative place and a collegial place. And so that brings all of these together in a mix that I really have not seen in other places. And I'm the old man here, so I can tell you I've seen more places.
I think immunology is a field that really needs technology. We've seen huge advances in our understanding of immunobiology through the advent of genetics, machine learning, computational approaches, and these are things that MIT is really good at. And especially in my field, we really need technology development to enable new discoveries. So I think that's a unique feature of doing immunology at MIT. We're at the cutting edge of technology that we can apply to our biological problems.
I don't have too much to add we haven't already heard. I'll just say I don't know if my lab could operate in the way it does at many other places in the world, because we need to be able to bridge experimental immunologists with computationalists and hybrids that can do both, and having access to those types of trainees, that's very uncommon, that want to do all three of those things at most other institutions.
So this, I think, is especially helpful for immunology, but it seems to be a property of MIT in general. It's pretty unique to combine fearlessness and rigor in a combination that actually works. Like the ability to try what might seem like it just definitely isn't going to work, or something that might seem a little bit crazy, but do it in a way where substance is never lost, where you make sure that we really are going to do this to make the impact, not just to make the flash in the pan. And I think that in immunology, everything my colleague said about technology and connections with each other, all of that's important. But I think that everybody from the sophomores and juniors in my lab who've started projects through to the graduate students, the postdocs, and my faculty colleagues, to combine fearlessness and rigor in one setting is a special thing.
Question over here.
Okay, I have two
You will get them.
Okay, I was a cheerleader, I could do this. Two questions. The first is, in the earlier session, they spoke about how difficult it was to move from a mouse model to a human model. And I'm wondering if that is less difficult in the work that you do, because it appears you're translating it faster and more readily. So that's question one. Question two is, I think for Professors Wong and Birbaum, is my understanding is that solid tumors are harder to treat these days. And is your work directed towards solid tumor treatment? So that's my question.
Okay. Maybe Facundo, you can start on the first question.
Those are excellent questions, and there is no perfect model, and every model helps to address a problem. A human clinical trial will take tens of millions of dollars and several years to plan. What we can take the advantage is in the mouse, for example, is to iterate things extremely fast and then narrow them down to be then tested in humans. And the technologies that we do in the lab that is trying to make this mouse more similar to humans are at the tip of this, because all the predictions, and there is an experiment ongoing, they are all the mouse experiments that we and many others have done. They have now done the human experiment and show that the predictions were absolutely right. So again, there is no perfect model. We will not know, everything will not work the same, but at least at a very diminished cost and time, we can operate in the mouse as a credible model.
And I think maybe to add specifically, because I was in the session, the immune system, I think, translates better because we're looking at systematic changes and not a single molecule in a pathway that has to be conserved between mouse and human. And for the second question, Michael and Murray.
Yeah. So for the second question, in terms of translation, it's a little bit of both. So the slide I showed towards the end is in models of melanoma, and so we're interested, which is a place where these tumor-infiltrating lymphocytes has been clinically approved, could stand to be improved. And so we really, even for mouse versus human, it's really sort of every tool for a job. So some of the questions we ask in terms of solid tumors where immunotherapy has been successful, some we're working in things like glioblastoma where progress of solid tumors has been slower. Some of it's in liquid tumors because that's where some of the clinical data is. And so we do our best to match depending on what we're working on.
Question over here and then back there.
Thank you. Thank you for the whole panel. Fantastic talk. So I have two questions actually. One for the whole panel. What do you think about mRNA technology? What are the advantages you see currently? Or do you see any limitations for your work you're doing in immunology for vaccines or maybe therapeutics? Second question is for Jessica. Love the talk, antibody lectins. Curious to see how you're going to apply this technology from oncology to autoimmune diseases. Thank you.
Sure, I'll start with the question directed at me. Thanks for that. So these applications and indications in autoimmunity and immunology that we are going after, in these indications, there's biological rationale for why you might want to target a glycoimmune checkpoint. So for example, in autoimmunity, while in cancer we block glycoimmune checkpoints to potentiate immune responses, in the context of autoimmunity, you could imagine engaging those same immune checkpoints to elicit tolerance in the same way that tumors tolerize the immune system to themselves. In the setting of fibrosis, there's a glycoimmune checkpoint that allows fibrosis to kind of continue unchecked that we are thinking about targeting to help slow that process down. But all of this builds upon fundamental biology, fundamental life science research that has shed light on these biological mechanisms.
Let's move over there.
Good afternoon, everyone. Stepping back a little bit, I was impressed with across a number of your presentations about just how dynamic the immune system is. Just the scale, the quantities that you're dealing with are just astronomical. And then on top of it, you have people. And so I was also impressed by the slide showing, if you could give 7 doses over 7 days with increasing, amounts, and you get a really tremendous response, but impractical. And I really want to target or push on that impracticality. I work with the state agency that really fights infectious disease, and I want to push back on that in practicality? What can we do to make that practical, or, so that's it.
No, I'm happy to address that. I mentioned that a bit too quickly in a brief talk like this, but we have now, in a very recent publication, which I flashed briefly, understanding that that is a practical challenge. We have now, by understanding the mechanisms better, been able to design a system which has only 2 shots, which takes it much closer to practicality, because 2 shots you can do. And so that's now slated to go to clinical trial. The 7th shot was already done, as I showed in humans, but that's where we are going with this, is to do the 2-shot regimen, because that does just as well, at least in mice.
Question over there.
Thank you for the exciting talk. I'd like to follow up a little bit on that question on probably a more fundamental question. In the 7 doses, I can understand that, like you're saying, the subdominant epitopes will increase, but viruses are equally smart. I'm just wondering, what if the previous epitope starts dominating again, because like viruses can mutate. You're trying to like decrease one epitope and increase the epitope. The second question, following up like with Hari's talk, he can actually like now visualize and quantify the timelines. Is it possible to, I don't know how different B-cell kinetics and T-cell kinetics are, but is it even possible to get both of them in the same timeline so that you can actually get both arms to have a good response?
So your questions are excellent, and to the part that I will address is the following, that I mentioned very briefly that we are trying to see how to leverage this sort of thinking into a potential cure for HIV. And what we are finding there is that when people have suppressed virus, but some amount there, then this may be happening. This is still very early days, but it may be happening that one by one, the epitopes get masked, and then you get more and more responses, and so on and so forth, while the virus is dormant. And so you can then create a net of antibodies that target different regions or epitopes, and as a consequence, when the virus starts to come up, escaping that net becomes very difficult. And so that's the part there. But I might also slightly comment on the second part of your question. In the extended dose delivery things that we have done, in order to optimize it to 2 doses such that it becomes closer to practicality, one of the things that happens is that you sync the T-cell and antibody B-cell responses to come more within synchrony. And if you don't give the adjuvant also in an extended way, then you won't let that to happen, because that's how you prime the innate immune cells, the dendritic cells, so that later on they're more efficient with the T-cell dynamics. So there has to be synchrony between those to get the best responses. Now, how best to image them and understand them better, I'll leave to Hari.
I'll just say that we and others have definitely shown that you can, similar to Arup's work with B cells, if you manipulate the dynamics of what the T cells are seeing, you can get better T cell responses. And we know that you can't get great B cell responses without great T cell responses. And I think it's just, we need to put it together. And actually, Facundo and Arup and I should probably work on something like that.
Can that is an excellent question. Antigen or virus is the fuel of this reaction. If that stays for a long time, this reaction goes on for a long time, and it goes on from B and T cell together. And Arup very elegantly showed that in these lymph nodes, when you just give 1 dose, you don't get antigen there for a prolonged period of time. So answering to your T cell question, that is very important, if you have the fuel that is the antigen there, you will keep the B cells and the T cells cooperating for a prolonged period. And if I make a comment with regards to, we know so little about vaccines. All we know is empirical, and even when we are giving a prime and a boost in humans, that has never been experimentally proved. The time between the prime and the boost is something that has been determined empirically. What we are trying to do, and what people do, is really understand the mechanisms for this in a way that we may alter this prime and boost to get a more effective immunoresponse. Exactly.
Perfect. I would have one last question I'm going to ask you guys to answer really, really quickly in the interest of time is, what is the next big frontier each of you want to tackle? I'm going to start with Michael for fairness.
It's going to be the integration with AI.
I think
Yeah. See, Stephanie said quick, and there's a timer counting down.
We can mingle after the panel.
It's I think, so in my world, it's being able to make general predictions and general designs in the same way that you can go to ChatGPT and make a picture of anything. Right now, the predictions are good but very narrow, very, very narrow. So it'd be the equivalent of having a ChatGPT that's really great at making pictures of ducks, and you ask it to make you a picture of a tiger, and it gives you a duck. So I think that getting to the point where some of these tools are going to be generally useful is something that we're really excited to work towards.
on my end, I think there are certainly biological questions we're very interested in, but on the technological side, you might have noticed that a lot of the dynamic live imaging that we're able to do, we're limited in how many cell types or readouts of function that we can look at, and there are practical limitations. So I think something we're really excited about and trying to think about is how can we visualize many, many more cell types and their states in real time in living organisms. And that's not a trivial challenge, but we have some ideas.
We are really focused on developing technologies that will allow us to kind of fit glycans into the broader picture of immunology and immune responses. And we need to understand this biology at a systems level and how it connects to all of the other aspects of molecular and cellular immunology that we understand. I think those kinds of fundamental advances are going to be necessary to translate this biology and to get to clinical advances.
In addition to the things I talked about, one of the things that we are launching in our group right now is a program to really understand how the innate and adaptive immune systems are integrated, because very little or much less is understood about that than anyone by themselves. And I believe that will get us to really start understanding questions like what determines the memory of an immune response? Why is it longer for one pathogen versus another? Why is it longer, perhaps, in protein antigens versus mRNA vaccines? And so that's one of the new frontiers that in our group is to bring that into innate and adaptive immunity together.
Yeah, I think the connection between quantitative immunology that is very recent and then translating that into computational or AI models is something that is extremely exciting and is going to be transformational over the last 10 to 20 years. I mean, we still know very little. We know we can generate antigens. We know we can generate antibodies. We can produce structures, but we cannot predict for better molecule binding. I think that the next 10 to 20 years are going to be transformational in that respect.
With that, I would like to thank the speakers and the panelists, and particularly you for your attention. Thank you.